This is a video for an HCI paper, but first, let's take a minute to review some plant biology. Plants are complex organisms with several different kinds of tissues. Roots, flowers, seeds, they're all made up of different kinds of cells. Each cell contains a complete record of the entire genome for the plant. A small plant, like Arabidopsis thaliana, has about 27,000 genes. Electronic fluorescent pictographs, also known as EFP images, are a widely used data visualization technique for identifying which genes are expressed where and under what circumstances. They display gene expression data on a diagram of the plant, with low expression levels colored yellow and high expression levels colored red. In order to identify which genes contribute to a particular biological function, researchers often examine many EFP images looking for red or yellow in one tissue or another. Consider the cognitive processes behind a similar task, a spot the differences game. The viewer directs his or her gaze to compare features between the two images. A faster way to find differences between the images would be to align them and then flip back and forth between them. This takes advantage of our ability for pre-attentive visual processing, our ability to recognize certain visual features in 250 milliseconds or less. Rapid serial visual presentation is a well-known method for displaying a set of images as a sequence viewed over time. It is widely known for reading applications, but the principle can also be applied to visual search tasks as well. Suppose you're looking for a gene that is only expressed in the root. RSVP makes it easy to see if a set of images contains one or more images that you're looking for. The question is, how do you control it? We compared 4 user interfaces to find out if RSVP can improve performance on visual search tasks using EFP images. 81 students from a bioinformatics methods course at the University of Toronto were randomly assigned to 4 groups. The first group was given a non-RSVP interface in which they had to manually select which image is displayed by pointing and clicking on the associated buttons. The second group was given a slideshow interface. Pressing a button at the bottom of the panel would start or stop a slideshow that displayed each of the images in a rapid sequence. The third group was given a hover interface. Hovering the mouse over the associated buttons would update the display accordingly. And the fourth group was given what we called a velocity control interface. This consisted of buttons to control the speed and direction of the RSVP display. Guess what we found? The effect of user interface method on task completion time was statistically significant. Post hoc tests showed significant differences between the hover and velocity groups and between the hover and point and click groups. The slideshow and velocity interfaces were not significantly faster than the point and click interface. As for accuracy, we found no significant differences between the 4 groups. It is nice to know that the improved performance in time does not come at the expense of accuracy. What does this mean? Well, we now have an empirical basis for including an RSVP display in our next generation of bioinformatics research tools. And while the slideshow and velocity control methods may be useful under certain circumstances, we found the hover interface to be the superior approach. Beyond the task investigated here, we believe that a hover RSVP interface can improve other visual search tasks as well. Gene identification, online shopping, photo cataloging, file management, email search. These are but a few applications that could benefit from this research. But that is work for another day. Thank you for watching.